Hi everyone, I am Saniya. This is cute little Yangshi. And in this video, you'll see these really cute dolphins in the back. And why am I showing this sea? Well, because in this video, I'll be talking about lncC, which is a really cool tool and a platform for long non-coding RNA related sets and enrichment analysis. So basically, if you have long non-coding RNA related sets and you want to do enrichment analysis of that, then this tool is for you. And it just came out. It just came out very recently. And I want to make this video especially for those of you like my subscriber, Juliet, who were asking how they could do enrichment analysis for these long non-coding RNAs, because those have been proven to play important roles in transcriptional processes and various biological functions. But we need tools to be able to understand if we have a list of non-coding RNAs, what do those do? How can we figure out their annotations and do enrichment analyses for these? So I was looking for a tool that could do this to help Juliet and other people out there like you who are watching this. And in the process, I found this tool that came from this paper here. And this is lncC. Yes, Yankee, let's dive into this lncC paper as well, and let's see some more about it. So basically, the tool that we'll be using comes from this paper here. So lncC, a platform for long non-coding RNA-related sets and enrichment analysis. And the database that they've created is freely available here at lnccindex.php here. So they talk about a lot of different things, which we will come back to, but they talk about how they collect reference set of lncRNAs as well here. You know, you can do searching, analysis, downloading, browsing, Yangshi, there's so much. You know, 18 types of reference sets, multiple functions like search, download, browse, enrichment analysis, ID conversion, ceRNA network, lncRNA expression, and statistics. There's quite a lot of information that we can see here about transcription factors binding to enhancers, chromatin accessibility promoters, microRNA, methylation, smORF, eQTLs. There's so much going on here as well. So in the paper, they talk all about like transcription factor type categories as well, other categories, other data sources. So it's a lot, how they can use this information there and how it's really helpful. So it's user-friendly data download. They were applying this for the Cancer Genome Atlas as well. And what we can see as a result of enrichment analysis as well. So there's quite a lot. And indeed, long non-coding RNAs. I'm going to talk through some examples here as well. So I'm going to take you through the tool that they mention, and then we'll come back and talk some more as well about long non-coding RNAs. But there are many different types of gene products that are out there as well. Before we dive into this more, is I want to talk to you more about what long non-coding RNAs are. Okay, so basically, so let me show that to you. So long non-coding RNAs are also called long ncRNAs or lncRNAs. They are a type of RNA that typically transcripts that are more than 200 nucleotides and they're not translated into protein. So nucleotides could be like A, T, G, C, you know, DNA nucleotides, and they are not translated into proteins. So being more than 200 nucleotides is sort of what separates these long non-coding RNAs from other types of small non-coding RNAs like microRNAs, small interfering RNAs, peri-interacting RNAs, small nucleolar RNAs, and other short RNAs. So long intervening intergenic non-coding RNAs, lncRNAs, are sequences of lncRNA which do not overlap with protein coding regions. So that's what lncRNAs are. So basically, when we look at long non-coding RNAs, they include intergenic lncRNAs, intronic non-coding RNAs, and sense and antisense lncRNAs. Each type has different positions along the DNA genome when in comparison and in relation to genes and exons. So this is a very helpful guide, which can show intergenic non-coding RNAs, which basically means that it's in between these two genes, A and B. So this is a non-coding RNA here in between, intergene, between genes. So this is an intronic long non-coding RNA. So basically what this means is that, hypothetically, if this was gene B, then this would be this long non-coding RNA is within this gene. So what happens, so it's an intron. So when we are forming the messenger RNA, then what happens is it's called, we can do alternative splicing, but basically exons are shoved together. Exons and introns are kicked out when we're making the final messenger RNA. So we get rid of the introns and we put the exons together. So introns typically aren't part of the final, protein product, or they're not part of the final messenger RNA that later on becomes a protein or polypeptide product. So basically these are long non-coding RNAs. They are intronic, so they're not part of the final messenger RNA, and they are between exons. This is antisense long non-coding RNA. So if transcription of, by transcription factors and RNA polymerase II of the gene is going in the 5' to 3' direction of the DNA, then this antisense lncRNA, AS lncRNA would be going in the other direction. And then this is bidirectional lncRNA. So, transcription is going in both directions. We can also have an enhancer RNA as well, eRNA, that's long non-coding RNA. We can have circular RNAs. So there's quite a bit of different types of RNAs available out there. So what I wanted to show you is basically some examples that I had created. So what I had done is I had collected some information on genes and various types of genes. So one of the things I had done here is I had kind of gone through a list of the different gene biotypes out there. And for this, I was looking at long non-coding RNAs, and I was just trying to select just a few random ones. We can see that there are quite a few different long non-coding RNAs. A lot of them are links as well, but they have various names out there. Zinc factors. But for simplicity, I want to show you guys this list here of 74. So basically I just randomly selected 74 of these long non-coding RNAs. And what we can see is if I take this here, I'm just going to copy these 74 long non-coding RNAs, and I'm going to go to the resource lncC here. So I'm going to go to, and I go to the lncC, I'll go here basically. And one thing I want to show you before we dive into this list of 74 genes and doing enrichment analysis for this is kind of what lncC is. You can see the C in the back of cute little dolphins. So it's a comprehensive human lncRNA sets resource and enrichment analysis platform. So it has more than 40,000 reference lncRNA sets that are involved in 18 categories: microRNA, drug, disease, methylation pattern, cancer-specific phenotype, linked RNA binding protein, cancer hallmark, subcellular localization, survival, linked RNA eQTL, cell marker enhancers, super enhancers, transcription factor, accessible chromatin, smORF, exosome, and conservation. Over 66 subcategories and 50,000 linked RNAs. They've looked at upstream transcription factors and DNA regulatory elements, looked at TF ChIP-seq, DNA-seq, ATAC-seq, and H327 acetylation ChIP-seq data associated with hundreds of human cell types. And they can get annotation and enrichment analysis functions of linker RNA sets associated with upstream regulators and downstream targets. So this lncC actually comes from this paper here, and it was just pushed out very recently, and it's lncC, a platform for long non-coding RNA related sets and enrichment analysis. And again, here's the link here, but they talk about how long non-coding RNAs have been proven to play important roles in transcriptional processes and various biological functions. So it's so important to have this collection of human, so this is for human long non-coding RNA sets, and it's urgent. So they can then do enrichment analysis for long non-coding RNAs. So it's important to know that these are not genes, these are long non-coding RNAs, but they still have a lot of functions out there. So lncC is a linked RNA sets database that can document a lot of resources for these human linked RNA sets. So much information here. They've looked at TFs, DNA regulatory elements, looked at TF transcription factor that helps, with the transcription of the gene into microRNA. Messenger RNA, sorry, not messenger RNA. DNA regulatory elements like, DNA-seq, ATAC-seq, regions of open, chromatin accessibility, H3K27 acetylation, like so histone 3, that DNA wraps around. This is all like epigenetic marks, ChIP-seq data, and it gives annotation and enrichment analysis of linked RNA sets associated with upstream regulators like transcription factors, downstream targets. And this platform provides a variety of types of linked RNA sets and supports lots of annotations and enrichment analysis. And again, this is how we can access this free database here. And they can be used as novel biomarkers. So single cell and other techniques have helped us identify the roles of long non-coding RNAs and what they could be doing, how they can be involved in various cancers, as well. So there's so much that this paper talks about here. They talk, and we can look at this again more, but they talk about, all these 18 types of reference sets that they are providing information on, like TF2 enhancer, chromatin accessibility promoter, eQTL, expression quantitative trait loci, microRNA methylation, a lot of different resources here, search, analysis, downloading. So it's pretty cool out there. So now let's go back to our list of 74 long non-coding RNAs. So the first thing I can show you is, well, we can either upload, we can do a file example, for instance. We can have an example file, or we can just like either upload a text file or some file, or we can just paste our list of these 74 long non-coding RNAs. And what we're gonna, so I pasted this list of these 74 here, and I'm gonna do convert. So one of the things we're gonna see that I was trying to highlight is that we get like, so I think it didn't quite find all of the 74, but it did find 52 out of the 74, which is kind of good. So it's giving us still some information on those. So for instance, for this link RNA here, it has identified the gene symbol and entry ID, and I want to show that those match, so link 00115. I had identified the entry ID of 79854 in my data set, and that is indeed what is found here by this lncC resource. And it also had aliases available and ensemble IDs and NCBI reference seq IDs. So you do get a lot of other information out here, which is also really helpful. So, there are various examples out there of all these results, and we can see all of these. And I just wanted to show you the example of how it can identify the entre ID and aliases and ensembles and NCBI reference seq IDs, which is really cool. There's also this help resource as well, where we can also see more about what this resource is doing. So, it provides a variety of types of these link sets for users and performs annotation and enrichment analysis. So you put in a link RNA list, and it looks at all these different types of data, and they can give you, p-value, false discovery rates, subclasses and sets and classes, and they have a lot of different lists of information that they are incorporating, which is really cool, like on diseases, drugs, microRNA, cancer phenotypes, enhancers and super enhancers, accessible chromatin, cell markers, subcellular localization, cancer hallmark, transcription factors, methylation patterns, RNA binding proteins, survival, smORF, exosome, eQTL, conservation. So quite a lot of different tools out there, which is really cool. So what I want to do next is I want to show you that we can do something that, so we can do some searching for our list of genes. We can see what those genes are involved in. We can do searching for our list of lncRNAs and see what those tend to be involved in. These are some statistics of these lncRNA sets, accessible chromatin, microRNA, survival. So we can enter in any of these identifiers that we want. So I'm going to put in again the 74 linked RNAs, but we can put in the gene symbol name, the ensemble ID, the NCBI reference seq, alias, entre gene ID, and then we can just start the search here. And one of the things in the meantime, while it's loading, is that they talk about how long non-coding RNAs are regulated by different regulatory elements like promoters, enhancers, and transcription factors, which bind to the regulatory regions. So they are looking at enhancers, super enhancers, accessible chromatin, transcription factor by collecting and processing large volumes of ChIP-seq, DNA-seq, ATAC-seq, and so on. So they have a lot that they're doing. So what's great is that, again, in the search, I can look up the annotations again for these long non-coding RNAs. I can also see for each of these individual link RNAs what they are, what diseases those are involved with. For instance, I could see this one is for Alzheimer's, link 01772. I can see the diseases that they are involved with as well, and typically what is the count of for glioma, for instance. I can check out glioma and see that there are 81 link RNAs in this set, and they'll give us information on where they got this from as well. But I can go back and see this is the count. Essentially what this count is telling us is this is the number of link RNAs in original set, biological set. So that is what this is telling us. That's what the count is saying. So the count of 81 is saying that there are 81 long non-coding RNAs in glioma. There are 52 long non-coding RNAs in pancreatic ductal adenocarcinoma. In Alzheimer's disease, there are 18 long non-coding RNAs that have so far been identified in this list here. We click this here. and Alzheimer's disease, then there are 18. So our long non-coding RNA is one of those, for instance. So our long non-coding RNA link 00339 is one of those 81 in glioma. And it is one of the ones in one of the 52 in pancreatic ductal adenocarcinoma, and one of the 147 in a non-small cell lung cancer. And both of these over here just happen to be 2 out of the 3. Also this one here, astrocytoma. So we can already see that quite a few are involved in astrocytoma. Quite a few are involved with that, which already has like 3,431. So quite a few different link RNAs are involved with that. So we can see the diseases that they are involved with and what subclasses. We can also, so again, count just tells you the total number in this set, in this biological set over here. That's what it's trying to tell you in this set here. What is the total number? And of course, each of these link RNAs that are identified out of our 74 will be there. If they are for their respective diseases, if there are any enrichments that have been found. We can also look up, drugs that are associated. And this also, again, the count will always tell you like how many link RNAs are associated with that set. And they tell you again how they got this lapatinib, for instance. You can always click this, which is really cool. And there's also microRNAs associated with the respective link RNAs, cancer phenotypes, enhancers. So yeah, this was a really cool tool that just came out like by Dr. Jaishin Chen and all of these doctors here in Nucleic Acids Research. It just came out, and it's on lncSeq. It's talking about this really cool tool that they have developed. Enhancers, the super enhancers, accessible chromatin. It's taking a little bit of time. Yeah, and this is like DNAs as well, and DNAs is DNAs is some endonuclease that so you can also see DNase I is an endonuclease that of the DNase family encoded by the human gene DNase 1. So a chromatin breakdown during apoptosis, cell death. It is important to prevent immune stimulation, and reduced activity may result in an increased risk for production of anti-nucleosome antibodies, a hallmark of SLE. There's quite a lot that goes on, but DNase is basically an endonuclease that helps a chromatin break down, like the DNA breakdown when the cell is going to die, and it does it in a way to protect us from worse effects. So this is a tax-seq tissue kidney. So subclass closest. So there's more going on, and there are also cell markers like this lymphoid primed multipotent progenitor cell. So lymphoids are pretty much they are producing lymphocytes and antibodies, immune cells basically. So lymphoid cells are any of the cells that mediate the production of immunity. So in the cell markers data set, there are just 5 link RNAs that are associated with this, with the lymphoid primed multipotent progenitor cell. So, cell markers, the data set is just like trying to identify what cell, these could be associated with some link RNAs are highly expressed, or they are markers for different cell types and tissues as well. Like, they could be like, hey, I'm only highly expressed in neurons, for instance, or just highly expressed in kidney cells, or in pancreatic cells, or B cells, or T cells, for instance, or various cells, or heart cells, right? So muscle cells, skin cells, so some of them could be very specific. So that's what clicking on these will give you more advice. So we just found this one just had 5 link RNAs, and link 01359 is one of those. There's also subcellular localization. There is cancer hallmark as well, prognosis of cancer. And this is transcription factors that are associated with the regulation of these linked RNAs. It's taking a little bit of time. You can see it's querying them below. Okay, so this set here, then they talk about this transcription factor as well, A549 June, MAX, MYC, SP1. These are like transcription factors that are associated with the link RNAs. So CTCF regulating link 00115, June, MYC as well. So this is like all a list of, transcription factors of ChIP-seq data of 467 sample types from ONCOBE, VMAP, Systrome, ChIP Atlas, and GTRD. The relationships between TFs and long non-coding RNAs are built via many kinds of link RNA-related regulatory regions like enhancers, promoters, chromatin accessibility regions bound by TFs. So you can already see this, that we can look at TFs binding to enhancer regions, TFs binding to chromatin accessibility regions, TFs binding to link RNA promoter regions as well. So this is a transcription factor, and for instance, June is a transcription factor. Protein coding gene. It's the first. Actually, it's one of the first oncogenic transcription factors discovered. Oncogenic means like it's sort of related to cancer as well, and it's a driver of cancer if it goes haywire. So yeah, it's kind of involved in quite a bit. So we can see all of these things. So we can maybe just look at maybe RNA binding proteins as well, but there's quite a lot here that you guys could see. And there's so many of these search results. So we could see RNA binding proteins, and what RNA binding proteins do is they bind to single or double-stranded RNA in cells. And participated in forming ribonucleoprotein complexes, RNA recognition motif, double-stranded RNA binding protein, zinc finger, and others as well. RNA binding protein involved in RNA editing events. So there's like so much more, and I'll try to make more videos on that, but this is the search. So basically what search does is it gives annotation results for genes in a gene list for each of the genes. If So that's what the search does. It gives us various biological annotation results for each of the link RNAs in a link RNA list. Sorry, it's just little out of habit for me. I usually do a lot of gene enrichment, but basically, so sorry, I've been messing up and saying this a lot, but basically it gives the various biological annotation results for each of the link RNAs in a link RNAs list, if annotations are available. So basically what that is doing is it's just kind of going through all of these different types, microRNA, drug, disease, annotation information, transcription factors, survival, smORF, and it's trying to just like give information about these each of these link RNAs if possible, and some survival. This is the Cancer Genome Atlas as well, and this is survival analysis, which is very key in biostatistics, doing survival analysis as well, univariate Cox regression analysis that is done on survival. So that's just for individual genes. Now, a key thing that we always like to do is if we have a list of, of link RNAs, like we have 74 link RNAs out here, we might want to do some enrichment analysis of those as well. We might want to see what those are involved with. So I can just, select these 74. And I can paste this here. And I think I want to keep the p-value at 0.05. I can look at link RNAs of link C or of GenCode. I'm going to just stick with the default of link C. I want to just select all of these. I'm going to just check all of them, and I'm going to have this run. So this here is doing link RNA set enrichment analysis. So that's what analysis is doing. This is basically link RNA set enrichment analysis. So for the set of 74 link RNAs, what do we find? That's what analysis is telling us. So we see that it's okay, 61.111%. So let's just wait for it to finish loading. So yay, awesome. So just for simplicity, I'm just going to move this here into a new thing. So we can see that we have the classes that have insignificant enrichments here: cancer phenotype, super enhancer, RNA binding protein, and survival. Those were certainly lists that we had looked at before, so we might look familiar as well. So what we see here is, these are sets like lung adenocarcinoma, never smoker versus lung adenocarcinoma normal. So what this one is doing is it's comparing one group with another. So it's comparing people who expression of the link RNA in people who never smoked, the 36 people who never smoked against 10 people who have, currently smoked. So it's a cancer RNA nexus that has 40 cancers and 325 phenotypes. And here, all they're doing in this type is they're comparing the expression of the link RNA between these two groups in this box plot. And basically, they had a list of those link RNAs. And in that set, they have identified 451 link RNAs in this site here. So link RNA number in the set is 451. And similarly, in this other one, we see that there's a cervical squamous cell carcinoma, so 305 compared with cervical squamous cell carcinoma adjacent normal, which is this. And there are 651 long non-coding RNAs in this set. And what they're trying to do here is we're doing something called an overlap analysis, where out of our 74, so we have 74 input lncRNAs. That's what we have here. And we're putting it into the enrichment analysis. So 6 out of our 74, were found in common with the 451 in the lung adenocarcinoma in this here in this set of 451. So we notice that this number is higher than 3, like 6 is higher than 3, and that this is smaller than this. So that means that the overlap here, 6 out of the 451 lncRNAs in this list were found in our set, as opposed to 3 out of the 651 that were found in this list. So already this, this first list is smaller, it's 451, and the overlap is higher. So the significance is even greater. So you notice that the p-value is smaller. So generally, the smaller the p-value, the more significant the results are. So we can say that the, lung adenocarcinoma one that we were looking at is more significant. It has smaller p-value, and that also smaller p-value reflects the significance of having 6 out of 451 lncRNAs in that set overlap with our set of 74 lncRNAs. So again, a higher overlap for a smaller list, 451 instead of 651. So smaller list and larger overlap makes it more significant. And we can indeed see the Venn diagram. We have 74 input. We have 6 that intersect. And this is the set of 451 in lung adenocarcinoma. So we have this 6 that intersect. So this is what this Venn diagram is doing, and that's what this Jaccard index is trying to calculate, the intersection of R74 with this 451 set, the 6, and seeing how significant that overlap is. And you'll notice that if we look at the other one here, the site is even bigger, and this is even smaller relative, and the intersection is 3, so not quite as significant. So we do some corrections, because we're doing multiple testing. Oftentimes what we do is we use multiple testing correction, like Benjamin Hochberg or Bonferroni corrections here. So that's what these are doing. These are correcting our p-value because we're doing multiple testing, and we don't want to have too many false positives, too many significant results that might have just been by chance, because we're repeating this again and again and again. As Einstein says, insanity is repeating the same thing multiple times, expecting a different outcome, I think. Anyways, so that just kind of shows you how this analysis works. What we could also do is we can save this. And if we save this, we can also note where it's saying that we have 6 that are in common. Here, we can also exactly see which 6 are in common here, like that it's like link 00115, this one, this one, this one, this one, this one. These are what is in common. So when we format it, it has the heading titles, and it reflects a little bit more than this here. We can also see for the second one, which are the 3 that were found, from our list with their list. So what we can see from there is like link 00337, 339, and 466. And again, this has the overlap of 3 out of this, and this one has 6 out of 451. Once you put it in like Excel, it might be a little bit needed than the text file, but saving it downloads that. You can also see a bubble chart here. One of the things I want to see and show you guys is a bar chart. Typically, they have negative log 10 p-values, and why we take negative log 10 p-values is because the p-value could be very, like on a very varying scale. As we can see from here, the p-value could really vary from like times 10 to the -5, and this one is not. So to put and to neatly plot the p-values on a nice scale that is not too stretched out. What we do is we often do the negative log 10 p-value. The negative's added because when you take log 10 of something between 0 and 1, it's negative. So negative log 10 p-value will just make it positive again. So that's what we observe here. And we would still expect that the lung adenocarcinoma would have a larger negative log 10 p-value. So it's more statistically significant. So again, smaller p-value, which is what we want, equals larger negative log 10 p-value. You have to be careful like that we consider the fact that these are not adjusted p-values, but it's still good because we have the original data that we can anyways use that has the corrections. But just to keep in mind, smaller p-value is larger negative log 10 p-value, and that's the same thing as being more significant. So again, because a lung adenocarcinoma is more significant, again, we expect this to be a little bit like 4.61. That's kind of more than 1.30. So we just have to plug in the p-value into the negative log 10 to get this result of 4.614. So that's what we can see for this as well. We can also look up the super enhancers and for this list of 74 as well, see the overlaps that are there, 11 out of 587. That's quite a lot of significance, for these super enhancers. And I'm not too sure about the super enhancers, but I think from the data set, it says that they have processed and collected this ChIP-seq data from NCBI, GEO, SRA, and Code Roadmap, GR. They look at 3 different positional relationships like overlap, proximal, and closest between enhancers and lncRNAs. And there are closest active lncRNAs with super enhancer identified by CRC. So there's quite a lot of more information here. And we can keep on doing some data browsing to see what all data sets do they have that are available here. It seems like quite a lot of information. You can look up all the sets that they have talked about, which is nice. We can also look up RNA binding proteins as well that are significantly, CERT6. Oh, that's interesting. It's a stress response protein deacetylase and mono ADP ribo encoded by the CERT6 gene, molecular related to aging. Yep, it's a sirtuin. That's what I figured. Sirtuin family of proteins. So it's related to aging as well. So that's interesting. RNA binding, and it's related to 51. And if you click this number, you can also see the lncRNAs that are associated with this enrichment class here of CERT6, which ones are involved here. Like for this EP300, which I think is another transcription factor that's key. You can see other, the 53 that are in common. And of course, you can also sort by count from smallest to greatest or the other way around, from largest to smallest. We could also look at like the survival one here too. And just like, I want to show you one more example that, like, let's say we have another list as well. So I just want to show you the results will differ based on this list here. So this is a list that has about 116 lncRNAs here again, but you can put in any of those as long as they are lncRNAs, and that's good. So p-value, I'm going to just make it back to 0.5 and then run this. Oh gosh, I need to do check all. So this is a list. Again, of 116. So meanwhile, I hope this is helpful, and while it's loading, I hope that similar to Juliet, if you have any questions, you can always reach out and ask me, and please subscribe to my channel. But I think that this is a really cool tool out there. So I'm hoping that this will be helpful for you guys in your work. This is a really neat tool that has just come out. So it's cutting edge. So now when I finish this here, again, we see that this lung adenocarcinoma seems to show up here, but now we have 7 that overlap, and we again can see these overlap here. We can see in the Venn diagram that now our input is 116 lncRNAs because the set that we put in is 116. The overlap is 7. And again, this remains the same at 451. So we just have an additional lncRNA there that, but we also still have to take into consideration that now our lncRNA set is bigger. It's 116. So the chances of having more hits, more commonalities, more intersections by chance is increased. So even though this is a higher number, we do have to discount it by the fact that, a little bit by the fact that we have a slightly bigger set. So that's how we have to do that. And then the cancer phenotype, that's what we saw for super enhancers. We see this here, RNA binding proteins, survival. And then last but not least, I want to show you a third example, and maybe this example of 85 lncRNAs. Let's just see what results this one will have. Let's see if we can get some more hits on this. I'm going to paste this list of 85, and I'm going to change this to 0.05. That's just my personal preference. And oh yeah, I keep forgetting to check all. And while it's loading, I want to show you how they do the enrichment analysis. When they find the p-value, they're kind of using this math to find this doing a hypergeometric test, essentially kind of looking at, our reference set versus the query list. The query list of lncRNAs that we've put in has a total of S lncRNAs, and then they have to look at the reference sets that have a total of N lncRNAs, and they have to run this sort of comparison, seeing how probable it is to just find that intersection by chance, or is there something significant about the number of lncRNAs that have intersected between your list and that biological reference list. If it's by chance, like what are the chances that you find that 7 out of 451 are found between the two lists. So that's a kind of like the math involved. And then there's like a Jack-Rid score, which is just looking at the similarity between the two sets. And there's a Simpson score as well, which is looking at the intersections in Venn diagrams. And I can prepare more videos on understanding these terms here. But now basically I gave it a last shot with this list of 85 different lncRNAs, and let's see if we can get some more interesting results. And it already seems like we are. Wow. So I can look again at the bubble chart as well. I can see, I have to download that, but I can see that this is, let me download this. I can already see this here. I can hover over it too. And you can always just take the data that they've given and you can just download it, and you can always just plot the data too. Once you load it in Excel or R, you can always plot the p-values and enrichment terms. And I can have other videos on those plottings. But you have like this cancer phenotype as well. There's quite a lot going on here. They even have even more results. So you see that it shows you the classes that are significant. If nothing showed up, then it won't show those to you. You can see enhancers, super enhancers, accessible chromatins, subcellular localization, cytoplasm, and nucleus, that's where those long non-coding RNAs are, like 23 of them on the cytoplasm, usually, and 14 of them on the nucleus. We can also look at transcription factors, RNA binding proteins, survival analysis as well. We can look at smORF. Let's see what the smORF looks like. Small, short, open reading frames in the, or is open reading frames in the body that can encode small peptides. So we can also look at eQTL, expression quantitative trait loci, which is sort of looking at how a SNP, a single nucleotide polymorphism, or one DNA base pair difference, how that can be if it's an A in one chromosome for one person, but it's a T in the corresponding chromosome for another individual instead. How does this one base nucleotide difference, how does this associate with changes in expression for this lncRNA? So they've looked at lncRNA eQTL, so genome-wide association studies. So there's just a lot that you can do. So I hope that this video would be helpful for you guys to see how to do lncRNA, enrichment analysis. So please let me know if you have any questions at all. I'm also kind of learning about lncRNAs, and I really hope that this is helpful. So I'm Saniya, and I hope that this is helpful for you guys. So please reach out if you have any questions at all. Thank you, and I hope to see you in the sea of opportunities in this field. Thank you, guys. So again, thank you guys for watching. So now I'm trying to blend in with this sea. You see the sea here and the sea in the background. So on behalf of Yangshi and me, thank you so much for watching this. It's in a nice little sea. Thank you guys for watching, and please do subscribe. Yangshi saying, please do subscribe.